Evrimin Dört Boyutu adlı kitabıyla evrimsel biyoloji alanındaki devrimin öncülerinden biri olarak kabul edilen Profesör Doktor Eva Jablonka'yı Evrimin Dört Boyutu, Genetik, Epigenetik, Davranışsal ve Sembolik Varyasyon adlı sunumunu gerçekleştirmek üzere kürsüye davet ediyoruz.
We would like to invite Professor Dr. Eva Jablonka to the stage. Jablonka currently works at the Tel Aviv University and is considered to be one of the revolutionaries of the field of evolutionary biology because of her and Marion Lamb's book Evolution in Four Dimensions. She will give us her presentation, namely Evolution in Four Dimensions.
Good afternoon. It's a very great pleasure for me to be here, and I'm very grateful to the organizers for inviting me. I want to I dedicate this lecture to a very dear friend of mine who died a short time ago. I want to thank the organizers, and also my friend Marion Lamb, with whom I worked and continue to work, and Dana Zhiligovsky, who did the pictures, to my students and my colleagues, who always contribute to everything that I do, and of course to you. And I just want to say that really for me coming here is a particularly big pleasure for several reasons. I will just mention three. The first reason is that it is a student organization that has invited me, and I think this is a wonderful initiative on your part, and I'm very, very happy to be part of it, because you are the future. It is for you that we work. You are going to continue and change whatever we are doing. The second reason is that I know that in Turkey evolution is under attack, and I come here as an act of solidarity, as a scientist, as an intellectual. We all belong to the same scientific world, to the same world of science. And it is in the name of this world that I'm coming here to talk to you about the science of evolution. And the third reason is that I was very happy when my book, Evolution in Four Dimensions, was translated into Turkish. And again, I want to express my gratitude. So thank you. So what I'm going to do here is I'm going to present a view of evolution that is very strongly influenced and informed by recent developments in developmental biology and especially in epigenetics. And I make several arguments. Some of them are critical arguments to some extent, but I want you to understand them in context. The first is, the critical part is, that I argue that the prevalent view, the common view, that we have to think about evolution as the evolution of genes and DNA is not wrong, it is correct, but it is too narrow. We cannot understand evolution fully if this is the only way that we think. So, what I'm saying is that the way that we have been thinking for the last 50 years or so, very, very much concentrating on the genetic aspects of life, of biology, is very important, but it is not sufficient. And because we're scientists, and because science progresses, and because we learn new things, we have to incorporate these new things into our theories of heredity, and into our theory of evolution. And this is what I will try to show you how I'm thinking about it. And the positive argument, what am I saying? What am I trying to say in this lecture? I'm trying to say that when we're thinking about the way that information is transmitted, it is transmitted through several channels. There are different types of variations that are heritable and that affect evolutionary change. And therefore, the way that we think about heredity and the way that we think about evolution has to be expanded. And I'm not alone in thinking that. There are many people today that are talking about an expanded evolutionary synthesis that needs to be done today in view of the new things that we have been learning. So let's come back to this picture. This picture has been drawn by my friend Anna Zieligowski, and it is called The Five Mothers. If we look at ourselves, each of us is different, right? Why are we different? What makes us different from each other? What are the reasons for the variability that we observe? So there are many reasons. We know that there are many reasons. First of all, we have different DNA. You see, this is the first mother. I have slightly different alleles from the alleles that you have, right? And this is a difference that makes a difference. This is an important difference. And it is part of what makes me what I am and what makes you what you are. But this is not the only thing. There are other differences that make a difference, that are important. What is in the cytoplasm of the egg is important and is influencing development. What is on the chromatin of chromosomes that we inherit from our parents is important for how we develop. The little RNAs that we get from both mother and father are very important for how we develop. All these things also makes us what we are. So it's not just the DNA, it's also the extra DNA things that are found in the sperm and in the egg. Then we're mammals. So we develop in uterus, right? The environment of the uterus is important and is influencing what we are. If, for example, our mother suffered during critical periods in pregnancy, starvation, this will affect us and our development. And not only us and our development, but it can also affect the development of our children, even if they grow in good conditions. And then there are, then there are things that happen after birth. The kind of milk that we drink, the food preferences that we develop depend on the traces in mother's milk. We get also immunological defense through the milk of our mother, and many other things. Not only food, not only energy, also information. And then there is the great mother, mother culture, that is so important for us human beings, that is making us also, to a very large extent, what we are. Our language, our culture, our traditions, all these things. Now what I'm saying now is self-evident. It is trivial. We all know this, right? What we didn't know, however, is that these things not only affect our development, how we develop, but they can affect heredity. We knew that the first mother, the DNA mother, can affect heredity. We didn't know that these things in the sperm, that come with the sperm and the egg can affect heredity for 1, 2, 3, sometimes many generations. We didn't know that what is happening in utero can have also transgenerational effect. And although we knew that what is happening in culture is transmitted from one generation to the next through cultural means, somehow we thought, well, this is not biology, this is culture. Why is it not biology? Everything is biology. The way that we understand things make changes in our brain. What we eat, how we eat, everything is entangled. We are living in a very, very rich biological world, which is affecting our development, and is affecting our heredity, and is therefore affecting our evolution. So I want to talk about this expanded view of evolution when we're taking into consideration all kinds of variations, all kinds of routes of transmission, and trying to understand the world, what we see, the diversity, the adaptation with these tools. It likes the picture. Oh, sorry. So this is another way of representing the same thing. I'm sorry. It's looking at what is happening through generations and what we're transmitting from one generation to the next. Organisms, genetic information, we're also transmitting the gametes, epigenetic information, also through the gametes. We're transmitting epigenetic information, also soma to soma, body to body, circumventing the gametic route. We are transmitting through behavior, traditions, things like that. We are transmitting niches that we construct. and in which our offspring and our descendants develop. And we have a very special way of transmitting information, we humans, and this is the symbolic way. We're transmitting information through symbols, through language. So I want to start with epigenetics. I'm not going to talk about genetics. I know you all studied genetics, you know about genetics, about DNA. It is part of basic biological knowledge. But not everyone knows very much about epigenetics. So I want to say a few words about it. And in the first place, I have to define the terms, because these terms have been used in many different ways. And there is confusion in the literature even now. So that's why it's important to start with definitions. So first of all, what is epigenetics? Epigenetics has a history. It's a term that was invented by Conrad Waddington in the late 1930s, in fact. But this term has undergone evolution, cultural evolution. It's not exactly the same as it was. Now we use it as the study of developmental processes in both prokaryotes and eukaryotes that lead to persistent, and this is very important, self-maintaining changes in the states of organisms, the components of organisms, or lineages of organisms. All right, so we're looking at things that lead to a change, but once this change happens, it can go on, it is persistent. And epigenetic inheritance is a component of epigenetics. It's not the same word. It is a specific part of epigenetics. Not every epigenetic mechanism leads to epigenetic inheritance. So epigenetic inheritance is a component of epigenetics, and it occurs when phenotypic variations that do not stem from variations in DNA base sequence are transmitted to subsequent generations. Variation can be transmitted in mitotically dividing lineages of cells, in lineage of organisms that reproduce asexually, and also in lineages of sexually reproducing organisms. And unlike the genetic systems, there are several ways in which this can happen. With DNA, we have a very complicated system with DNA polymerase that copies DNA. And we have cell division, and we have mitosis or meiosis, and there is inheritance of the sequence or variation in the sequence if there is mutation or a combination or any other events that changes it. With epigenetic mechanisms, we have several classes of mechanisms, and I want just to introduce these mechanisms to you so that we can understand how they work, what their limitations are, and what they can lead to. So we're starting looking at cells and what happens in cells that leads to epigenetic inheritance. Now remember, we're not changing the DNA. The DNA stays exactly the same. So what do we have? We have several mechanisms, and I will tell you about each of them very little. Some of them most of you know, but maybe not from exactly this point of view. So there are mechanisms that sustain very particular states of the cell through self-sustaining loops, metabolic loops. There are mechanisms that depends on the three-dimensional templating of biochemical structures. There are chromatin marks, and there is a whole zoo of chromatin marks. There are many different types of chromatin marks. And there are small RNAs, which are very, very hot topic today, as you probably all know very well. And these mechanisms are operating during cell division in prokaryotes, mitotic cell division in the soma, in the body of eukaryotic organisms, and also sometimes during meiosis. So there is transmission through meiosis of epigenetic variations. Right, here is a very simple example of a self-sustaining loop, which I want to show. Now, what we have to imagine, let's think about some microorganisms. One, unicellular, okay? Unicellular organisms. We have two populations of unicellular organisms. They live in exactly the same environment. They have exactly the same DNA, but they have different phenotypes. Why? They have the same DNA. They have the same environment. What's going on? Well, what's going on is something to do with the history of these organisms. Once upon a time, in one of these populations, this gene, there was a stimulus which changed the activity of the gene. You see, the gene is the one which has a, oops, so sorry, controlling it there. Okay, the gene has here a blue region, which is control region, and the light blue region, which is a coding region. And the stimulus came and attached itself here to the regulatory region. As a result of that, we had some kind of activation. There was a gene product. This gene product, however, had an interesting effect. What it did was, in addition to changing the phenotype of the cell, it also is a regulatory element, which is a positive enhancer of this gene. And therefore, this, once it attaches itself here, the gene is active again. So even when we don't have the stimulus, because we already have the gene product enhances the activity of the cell. As a result of this, more product is produced, the product attaches itself to the regulatory region, and so on and so on. So we don't need the stimulus anymore, because we have internalized it as the product of the gene. So when we're looking here at this population and this population, the DNA is the same, the environment is the same. But because of the history of this population, we have different phenotype here and different phenotype here. And this is an example of an epigenetic variation. It is a variation that does not depend on DNA-based sequence. It depends on the activity pattern of ancestors. This is an example, and there are many examples of this type of thing in microorganisms. One example has been described in great detail in fungus, in Candida albicans. It's a fungus that some of you may know because it's a nasty fungus that causes all kinds of problems, pathologies. And this Candida albicans can exist in two forms, opaque and white, and it can switch between these two forms, and it can be 200 generation in one state and 200 generation in another state. It's the same DNA, it's the same environment, nevertheless the epigenetic state of the cell is very different. So this is an example, and there are many other examples. Now the other example, this is one type of mechanism. The other type of mechanism that is very important depends on three-dimensional templating. And here what we have, and the best known example, it's not the only example, but the best known example is that of prions. Now what happens with prions is that we can have a cell, again think about a yeast cell, a unicellular organism, a yeast cell. In this yeast cell we can have a protein which has, in the two cells you have the same protein, the same amino acid sequence. What is different is the way that they form a three-dimensional structure. And in one case they form a normal structure, in a second case they form a prion structure. And the prion structure has a very interesting property, which we see here. Here is the prion, here is the normal structure of the protein. Now, as I said, the amino acid sequence is the same. It's the way that they sort of organize themselves in space that is different. However, once a prion meets with the normal protein, which is produced by the cell, there is a kind of binding between them, and it changes the normal protein into its own image. Right? It transforms it into its own image. And you know that there are all kinds of diseases like the mad cow disease, BSE, and at least 20 degenerative diseases of the nervous system that are based on prions. But prions not only do bad things for us, they're also part of normal development. And apparently can have adaptive functions, for example in yeast. There was a recent paper in Science, like a month ago, where many prions of yeast were described, and it was shown that in some conditions having a prion is a good thing for the yeast. It helps it to cope with all kinds of adverse conditions. Now, it's very interesting because what we have again, we can look at two yeast cells. One has a prion protein, the other one has a normal protein. DNA is the same, conditions are the same, yet the phenotype is different. There can be switches from one to the other. The switches can be spontaneous, or they can be induced to some extent by the environment. So if you are in certain types of stress, the probability of making a prion is higher. Okay. And here is this example with yeast and which has two types of proteins, one prion protein, one normal protein. As a result of phenotypes, they have adapted the environments. And a prion can be quite stable. It can change in like 10 to the minus 5 the rate, which is not as high as, not as low as the rate of the classical mutation, but it is quite low rate of change. So it can persist in the population for a long time. And if there is also selection for the stability of this phenotype, then any mutation in DNA that is leading to increased stability will be selected, of course. Now, the best understood type of epigenetic system, the one that we, first of all, is very, very intensely researched by many groups, and is also important for epigenetic inheritance, both in plants and in animals, and in sexually reproducing multicellular organisms like ourselves, are those related to chromatin. Now, I will not go into all the details in this complicated slide, but as you know, DNA is only naked DNA in the lab. Normally, in a cell, it is wrapped around the histone core, and lots of proteins are sitting on it. It is in many, many organisms. It has all kinds of modifications, such as DNA, such as methylation of the cytosine that is next door to guanosine, what is called DNA methylation. The histones can be different, and the histones can have all kinds of chemical modifications, which are very, very important, deciding whether or not this piece of DNA will be transcribed or not transcribed. Okay? So we can have the same piece of DNA, let's say in different cells. The chromatin can be different, like in a liver cell and in a skin cell. A particular DNA sequence can have very different chromatin, and the state of this chromatin can be very important in determining whether or not this piece of DNA will be transcribed or not transcribed. More than that, there are mechanisms that ensure that not only is a state of activity on or off, but also that the state of activity is inherited in cell lineages. So, for example, if you switch or change the chromatin state of a cell, this can be transmitted through mitosis, from one cell generation to the next, from one cell generation to the next. And I want to show you an example of this with a system that we understand best at the mechanistic level with DNA methylation. So here is the same piece of DNA. We have on the upper side and on the lower side, the up bit and the bottom bit have exactly the same DNA. The difference, and there are 3 CG sites on this DNA. In the upper bit, 2 of them are methylated. You can see them, right? Here, and here, here, and here. And notice that CG is always opposite it is GC. CG, GC, right? So it's a mirror image, it's a palindrome. Now what happens during replication is, this is the old strand, and this is the new strand. Right? The new strand doesn't have methylation. But there is an enzyme called methyltransferase. And when it sees a symmetry like this, here, and like this here, it sticks a methyl on the strand that is not methylated. It doesn't care about this strand here, because it's symmetrical. It only cares about asymmetrical strand. So look here, this is the same DNA methylation. Here we have this site methylated, this are not methylated. Here we have DNA replication. It's only here and here that we have the asymmetrical situation. So the methyltransferase sticks a methyl here and a methyl here, right? So what is happening is that we have a reconstruction of the methylation pattern, okay? So we started with this methylation pattern and we end with this methylation pattern. We started with this and we end with this. It's a memory system. It's a heredity system that is, in a way, here, hitchhiking on the semi-conservative replication of DNA. But it's not semi-replication, but it's independent of the sequence of DNA because the sequence is exactly the same, okay? Right. Now, let us go, let us continue. Okay, very nice. What does it mean? First of all, I have to tell you one thing. The important thing is that the method, if a region, in a control region, in a promoter, is heavily methylated, the gene is inactive usually. There are many qualifications to this, but this is the general picture. If the same region is unmethylated, the gene has a high probability of being active. So the pattern of methylation is important not only as a heredity system that doesn't have phenotypic consequences, it does have phenotypic consequences. Or at least it is part of a system that can lead to phenotypic consequences. Okay? Now, okay, you can say very nice, very interesting, but how, is it inherited between generations? And the answer is yes. And we have today many examples. One of the nice examples is in a plant, was found in a plant. This is Peloria. This is a plant called Linaria vulgaris. And what we see here, just a minute, let me find it. Right. What we see here, this is the normal plant, the wild type, this is the abnormal plant. And for a long time it was thought that this is a mutation. And people wanted to understand where this mutation came from, and now we understand quite a lot about the genetics of flowering. So people thought, well, let's have a look. And the reason they wanted to have a look is that because this flower has a very interesting history. The history of this flower is that when Linnaeus in the 18th century found this flower, he was deeply shocked. He realized that it's the same species as this, but he believed that the flower is indicative of a species, is defining a species. And he said, this is like a cow that is being born with a wolf's head. It was a terrible thing, and it shook his, he was a very religious man, it shook his religious convictions that God created everything in 6 days. And he said, well, God did create everything, but not in 6 days. At first he created all kinds of genera, and then he sort of, through hybridization or other processes, this genera evolved into the species that we now see. So this flower is important in the history of biology, and that's why people wanted to work on it. Because it was sort of a sexy kind of project. So they worked, and they looked at the gene. There was no difference. It was exactly the same here and exactly the same here. So what the hell is going on? So they looked at methylation, and the methylation was different. The methylation here was reduced relative to this in some regions. And this was transmitted from one generation to the next. Okay? So the difference between these two flowers is epigenetic and is inherited. And there are many other examples of this in plants. This is a recent paper about a plant called the monkey flower. And in the monkey flower we can have two types. One type without trichomes and one type with trichomes. Trichomes are very thin hairs that appear on the leaves, and they are a defense mechanism against grazing. So animals that graze, they don't like plants with trichomes. And never mind about exactly how the experiment was done, but what was found was that this difference is due not to a genetic difference, but again to an epigenetic, inherited difference. So having a trichomes or not having trichomes, which is an adaptive trait, and this is the point of this case, it is adaptive. It's not just a variation, maybe a pathological variation. It is an adaptive variation, and it is based on an epigenetic difference. And here I gave you a quotation from a recent paper in Science that has looked at lines of plants that were, you know, sort of continued for 30 generations. And today we have the ability to look at methylation really at a very, very high resolution. We can look at every methylation site in the DNA, and we can say whether this site is inherited or is not inherited from one generation to the next. And we can find out a lot of things about this. So you can think, well, maybe the stories that I'm telling you, it's really one example, one small example, something abnormal that happens in somewhere in the plant. But what we now know is, no, it isn't. Oops, sorry. There are 1114-287 CG site polymorphisms in Arabidopsis, in this line of Arabidopsis thaliana. It's a lot. It's more than 100,000 sites are different. And when you are looking at regions, you have big regions, right, that have a difference. You also see that it's 2,485 differentially methylated regions. And the rate of change is something like 3 times 10 to the minus 4. So it changes. In different regions, it changes at a different rate. And it doesn't change every generation, right? It can be transmitted. And things can accumulate if they are selectively advantageous. So here we have a very, very clear example of a very potent and ubiquitous inheritance system, because there's nothing very special about Arabidopsis thaliana. You know, it's a plant. There are many other plants that are like this, and they're all methylated. Now, we don't know about each and every one of them, and there are little quirks, and I'm sure that we'll find very interesting things about other plants. But basically, I believe that the picture that we have about Arabidopsis thaliana is a general picture. So you can say, well, very well. What about animals? We have been talking about plants. So here are 2 animals, 2 mice. And they look rather different, don't they? Yeah? One big, fat, yellow, and diabetic, by the way, and have a tendency to develop all kinds of nasty tumors. And the other one, a normal little brown mouse. Normal, thin, you know, not normal. And they have the same DNA. Exactly the same DNA. They are very different. Why are they different? Because of what happened to their mothers. It's a particular strain of mice. Again, I will not go into details. In this particular strain of mice, there is a potential for developing this phenotype, but this phenotype will not develop if the mother is given during her pregnancy folic acid, which is a methyl donor. Moreover, if we will look at this yellow fat mice and this brown mice, and we look at their offspring, this mice will have more yellow offspring than this mouse. This mouse will have more brown offspring than this mouse, right? So this is again transmitted in a complicated way, but it is transmitted. Now I want to tell you another story about methylation, and this time about something which I think is relevant to all of us, not only to the rats in which this work was done. You know that there are all kinds of chemicals in this world that are estrogen or hormone disruptors of androgens or other kinds of hormones, and we use this in agriculture, for example, for spraying vines. For spraying vines, we use kind of stuff called inclozoline, which is an androgen suppressor. And so we find this kind of androgen suppressors in fungicides, in pesticides, all kinds of plastics, in many, many things. And we know already that if this kind of stuff is found in the water, it can affect the development of animals. It can cause all kinds of problems with sex determination, for example. And all kinds of diseases can develop. Okay, we know that. Very sad, but that didn't make it didn't influence the big companies that are making this stuff. Now, an important experiment was done in 2005, and in that experiment, Michael Skinner took rats and injected a pregnant rat with this vinclozolin. And he injected her with the vinclozolin here, okay, and he looked to see what will happen to her male offspring and to the offspring of the male offspring and so on. He looked at the male offspring not because the females are not affected, but because the females can transmit information in many ways, not only through chromatin, but also through the cytoplasm and through womb and through behavior and so on. Whereas the males, what they give is the sperm with the genetic and epigenetic informations on this sperm, and that's all. So that's why he was interested in the males. So here we have, the males are abnormal. They have all kinds of problems. 90% of them have problems. And there is no selection, by the way. He checked for that. It's not a selection process. There's no selection among the males. They are sick. Okay, so you take a sick, this male and you make it with a normal finger, but never saw this vinclozolin. He also was exposed in utero. You look at their offspring, male offspring again, you see the problem. Then you look another generation, you again see the problem. For 4 generations, this disease was passed on in the male lineage. And he was looking also at the methylation, and he saw changes in methylation in thousands of genes. So this is what we had. So, you know, the amounts that he was using here were non-physiological. They were high. But we know that sperm counts in the whole world of males is going down. This is one of the things that this kind of stuff is causing. And I think that we have to be take this result very, very serious. And it's not just this result. I mean, you know about bisphenol A. It's a kind of chemical that is present in many, many plastics to make them tough. Bisphenol A is known also to cause pregnant women all kinds of problems to their offspring, cervical cancer, for example. And a very recent experiment that was published a month ago used physiological concentrations of this bisphenol A in mice and showed that it is transmitted up to the 6th generation. You know, we really have to think very hard what we are doing, because it's not only affecting us, it's not only affecting our children, it's affecting generation later. We are really ruining the developmental robustness of our descendants if we don't take it seriously and not insist on good epidemiological studies and on good studies that will look at the long-term effects of this kind of chemicals. Okay. Now, this is not the only thing this is not the only type of here you have, by the way, examples of normal seminiferous tubers from a normal mouse, which is normal sperm, and that's how the sperm of a vinclozolin affected mouse, rat looks. Not very good. Okay, I want to talk a little bit, because I guess that you all know quite a lot about small RNAs. You know that small RNAs are very, very important regulators of gene activity. Not only are they regulators of gene activity, but they can be also transmitted from one generation to the next. In the last Science and Nature papers, there are 4 papers from Mello and Fire Labs, the people who got the Nobel Prize for their discovery of small RNAs in 2006, that are describing the transgenerational long-term effects of small RNA in C. elegans. But it's not only in C. elegans. This is an example of injecting small RNAs. A small RNA, it is a silencer, right? It silences genes, and there was a small RNA was injected into oocytes, and this small RNA sort of silenced a gene which is involved in the development of the heart, and this was transmitted for 2 generations in these mice. How exactly? We don't know the details. We're learning about it. These new papers in Science and Nature about C. elegans are beginning to give us some clues. It seems that what the small RNAs are doing from these papers is they are changing the chromatin. And it is the chromatin not the small RNAs, that is inherited. Right? This is how it works. So the inheritance systems, the chromatin inheritance system and the small RNA inheritance systems are related to each other and they work together. Okay, now, because when I was talking about these things, I was talking for many years about epigenetic inheritance, because before it became a popular subject. And in the old days, people were always telling me, yes, you can find an example here, you find an example there. How many examples are there? So I did a big review with a student of mine, and we found many examples. Over 102 cases of epigenetic inheritance, which were found not because people looked for it, just as a byproduct of another research. And this 102 cases involved many, many genes, not just one locus, sometimes many loci, and it was not a completely full picture, of course, first of all because we missed some cases, inevitably, but also because we didn't include cases which were very similar, right? More or less exactly the same, just in a slightly different species. We didn't include this. But this is now, in a way, this work that I did is to some extent unnecessary because of the work that I told you about Arabidopsis. If you have in one plant over 100,000 polymorphic sites of CPGs, well, I don't need to say anymore. This is a very important phenomenon because DNA methylation is very, very prevalent in plants, in animals, in fungi, even in bacteria, by the way, and so on. And it's important as part of what regulates gene activity. Now, okay, this is all very nice. So we know that we have a way of changing in a persistent way that is creating new variations that can be transmitted between generations. What is the effect of it? What are the evolutionary consequences of such a thing? How can we think about it? We're very, very used to think about DNA. But we have to think about other things. What is going on? What can happen? Now, one of the interesting things about epigenetic systems is that they are both heredity systems. There is a mechanism of transmitting the variation, but they are also part of development. They are response systems. And especially they are response systems when there are stresses. So here we have an example of what can happen. Here you have, there are all kinds of stresses that can happen to a plant. For example, there can be a drought. It doesn't have enough water, it has too much sun, it doesn't have enough nutrients, and so on. There can be a bottleneck which changes the genetic constitution and leads to a very high level of homozygosity. This is a genomic stress. Another type of genomic stress is polyploidy, when you double the number of chromosomes, or hybridization. All these things, whether they are ecological or whether they are genetic stresses, what they do is they create a lot of epigenetic changes within the genome. So the epigenome is changed. There is a lot of epigenetic variation appearing as a result of this stress. And because this is inherited variation, selection can act on it. So exactly at the periods when we need it most, which are the periods of stress, we have a burst of variation. It's not genetic, it's epigenetic. There is also genetic variation, by the way, because transposons begin to jump, for example. And transposons, interestingly, are silenced by small RNAs and by methylation. So all this genetic and epigenetic are also related. But we shall not go into this complication, although this is very important. So if you're just thinking about the epigenetic aspect, what we have is an increase in epigenetic variation exactly at the time when we most need it, which is the time of stress, and then selection can work and lead to epigenetic adaptations, which later can be genetically assimilated or not. Okay? And here we have an example. There was very nice work in Israel on synthetic wheat. You can reconstruct the wheat because we know what kind of hybridizations and polyploidization events were involved. And you can look in the lab, do the experiment again and again and again and again, and look at what happens, not only at the genetic level, but also at the epigenetic level. And what they found was a huge amount of epigenetic changes. 13%, 13% of the methylation sites change their methylation status. It's a lot. And then you have this period of, like cows, for 5 generations, and then it begins to stabilize. And afterwards it behaved nicely, like classical evolution. But you have a kind of revolutionary period, they call it. Revolutionary epigenetic period. Also, to some extent, genetic. So, this is one way of thinking. Now, what I was talking to you about until now, I don't know how long I have, you have to stop me. Okay, so I was talking to you now about things that can be transmitted through the gametes, right? All these things are transmitted through the gametes. Chromatin marks, methylation, the methylation patterns, the small RNAs that I was talking about are transmitted through the gametes. But not everything needs to be transmitted through the gametes, because we can have changes that are induced, that are leading to phenotypic changes, not affecting the gametes, and nevertheless are transmitted from one generation to the next. How? There are many, many ways of doing this. Many, many, many mechanisms. And people are very interested in this mechanism today because they are very important for medicine, of course. And also for evolution. In fact, if we are wanting to think about evolution in a kind of more realistic sense, we have to take this type of mechanisms into consideration. So we can transmit from one generation to the next symbionts and parasites. We can transmit all kinds of products of development. Hormonal states can be reconstructed from one generation to the next. We can change morphologies even in such a way that is reconstructed again in the next generation. And in the next generation, for example, a woman with a small womb will have a small daughter, who also will have a small womb, who also will have a small daughter, and so on. So there will be a kind of transmission of constraints in this way. But it's just an example. There are many, many ways of transmitting all kinds of phenotypic states from one generation to the next without changing the DNA at all. And we can also change the ecology in which we live, which in turn changes the way that we develop, we and our offspring. So this is changes that are called niche construction. We construct the niches in which we live, in which we are selected, and in which we develop, and in which our descendants are also selected and are developed. And they reconstruct these niches through their own behavior. So there are many ways of transmitting information which bypasses the germline. Here is a very well-known example and very beautiful example of something that is happening in the rat. Mother rat is licking her offspring and sitting over them, right? And giving them warmth. And how much she does it, how much licking she gives to the offspring is very important for their development. The licking is not only keeping them clean, of course this is important, but you don't need so much licking to keep them clean. What the licking is doing is it's leading to a better maturation of the nervous system. And if you're looking at well-licked offspring, and offspring that were licked, they were hygienic, but they were not licked a lot, you see that they behave differently. The ones that were given a lot of loving care and licking, when they grow up, they become brave little rats. They sort of look at the environment, they are not afraid. They are afraid, but not very much. They are exploratory, and so on and so forth. And they are not stressed very easily. Whereas rats that were not licked a lot are very, very neurotic. Yes, they are afraid of everything. And we know that this is not something genetic because we can do cross-fostering experiments. We can take the rats from a well licker to a bad licker, from a bad licker to a well licker, to a good licker, and we can see what happens. And what happens depends on the behavior of the mother. It doesn't matter the genetics of the mother. Now, this is also transmitted from one generation to the next. Why? Because the offspring of a good licker mother, the daughters, become good lickers. And the offspring of a bad licker mother become bad lickers. So this is transmitted in the lineage. Well, this was known, by the way, for a long time, because the psychologists were working on this type of things for ages, and they knew it. But there was no molecular mechanism. So they were working on their things, the molecular biologists were working on their things, and they never talked together. One of the important things that happened during the beginning of the 21st century is that people started to talk together and to try to understand what kind of mechanisms are involved in this kind of thing. And they found that in this case, for example, what is happening is that we have changes in, by the way, many genes, but we're now focusing of the glucocorticoid gene in the rat. And what we're seeing is that when we have high LG, leaking and grooming, we have the GR gene activity is going up. As a result of this, the GR receptors are also going up, and the stress response is down. And the opposite happens in the bad lipids. Now, what is happening there? This is happening in the brain, by the way. And they looked at the GR gene to see what is happening to it, and guess what happened? The methylation changed here, on this particular site. And you can see a very consistent difference in the methylation pattern in good liquors versus bad liquors. Now, although a methylation mechanism is in work here, this is not transmitted through the gametes. Remember, this is in the brain, right? It is transmitted by reconstructing the development of the mother in her daughters. Because the mother has a mother that doesn't lick her offspring properly, leads to the development of a daughter who is neurotic. The neurotic daughter doesn't lick her offspring properly as part of her neurosis. And so it goes. And by the way, a very similar phenomenon is found also in human beings. Also, there is a psychopathology in human beings which was thought to be genetic and has been found to be due to the lack of touch of certain psychotic mothers of their offspring. They didn't hold them, they didn't give them the warmth and the caressing that children need. Children don't need just milk and to be clean. Children need to be touched. And they were not touched. And if they are not touched for the first 6 weeks of life, a psychopathology develops. A very, very bad psychopathology. We can see it especially in orphanages sometimes. Okay. Now, there are other ways of transmitting information. You can transmit information through behavioral means. In birds, for example. There are song dialects, and they can be transmitted, and they can evolve. They can be different in different populations, and they can even lead to sexual preferences. A female from population A doesn't like the song of a male from population B. It's not the song she heard from her daddy. She liked the song of her father or her neighbors, not any other song. And this can even create, in some cases, the beginning of reproductive isolation, which is the beginning of speciation. And there is a very famous story of the tits, in the 1920s, discovered people used to put milk bottles with a cap on them near the doors of, in England and in Europe, right? Now the tits discovered that if they flip the top away, they can eat the rich cream. And very quickly, we don't know who discovered it, who was the tit, who was the genius, the Einstein, who discovered this thing. But what we know is that once it discovered, it was like an epidemic. It started going on in the whole population of the tits in England, not only in England, but also in other countries. It crossed the channel, and it also affected 11 other species of birds, right? So here we have an example of cultural evolution in birds, which is very rapid, and, well, it was very good for them. And you know, what happens with children, they invent new things all the time and new variations of all kinds of things. We invent things all the time. This is just a small example from the kindergarten. But it's not just us, it's also our close relatives. There's more and more work on chimpanzee cultures, and we know that different populations of chimpanzees, for example, can have different ways of dealing with the environment, cracking nuts, hunting termites, hunting red colobus monkeys, finding water, doing all kinds of very, very interesting things. There are all the time more and more and more examples. There was a famous paper in 1999 where 39 different traditions were found in Africa, but there are many more now already. And it's a very long project because it requires a lot of collaborative work and long-term work, and it's difficult to find grant money for this kind of things. But nevertheless, brave people are doing it, and there are more and more traditions being found, not only in chimpanzees, not only in gorillas, not only in the great apes, but also in dolphins, also in all kinds of other animals, and you will be surprised there are also differences between populations of invertebrates that are due not to genetic changes, but to changes in which behavior is being reconstructed from one generation to the next without genetic changes. So, now, how important is it for evolution? I don't have time to go into all the evolutionary ramifications of these things. I was trying to do it, you know, I need a time to present a whole book for this. That's why I'm writing books. Yeah? But it is clear that if we have variation that can be transmitted from one generation to the next, this can lead to cumulative evolution. It can have effects. It can lead to, it can interact. The different inheritance systems can interact. For example, if you select for a cultural change, for example, for the domestication of cows, we have domesticated the cows, we started to drink milk. We started to drink milk, there was an advantage for people who can break down lactose, right? So we created a cultural environment which was transmitted from one generation to the next, which has a selective effect on genes. And that's why there are, in populations that have used milk as a source of energy, the allele for breaking up lactose is high, whereas in other populations it is low. So here we are. And what can we say in general as a conclusion? What we can say is that we do not have this kind of division of label between development and heredity that we used to have in the old way of thinking. We had heredity and you had development. And what happens in development doesn't affect what happens in heredity. These are two different channels. It doesn't affect it directly. But this is not correct. We can have a soft inheritance. This is the inheritance of developmental variations. It can occur within populations. And it can occur on the epigenetic axis. Right? This is the first thing. The second thing is that when we are looking at the genome and at the epigenome, we don't think about it just as genes. There are lots of genes. This gene, this gene, this gene. It's a system. And we have to think about it as a developmental system. As a system, as an organ of the cell, as Barbara McClintock had said many, many years ago. And she's right, I think. So we have to think about the genome and the epigenome as a developmental system, which has its own variational mechanisms. There are mechanisms which have been probably honed by selection for producing variations in the epigenome. It is very naive to think that the only thing that did not undergo any evolution is the variational system. Why? Everything else undergoes evolution, and the system that leads to variation cannot do it? Of course it can. And therefore, we must understand that what we call Lamarckian mechanism, this is developmental mechanisms, that allow this developmental inheritance are important in both microevolution, populations, and they can also lead to macroevolutionary changes, because we know that during stress, there is a huge burst of variation in many loci, in a coordinated manner. And this can be then selected. So it's not a small change in one of 10 to the 9th bases of DNA that is important. That can be also, well, I'm not saying it's not important. I'm saying this is not the whole story. We also have this mechanism that can lead to very big saltational changes that can be very important in evolution. Thank you very much.
hata sonucu açısından önemli olan orada vurgulanan doğal seçilimin ne kadar güçlü olduğudur.